Just hold on. Okay, that's right. Okay, so as I said, first we're gonna give you an overview. So we're gonna show you some aspects of the practical work. So we need to work in a hood and in aseptic conditions, and also then we'll go through some of the culture parameters. So usually cell culture work, it's done in a biological safety cabinet, which essentially has laminar flow. And that means that it has a constant flow of air that basically prevents from airborne organisms to entering the hood. Sometimes we also have this closed cabinet, so that also can protect us, depending what type of cell culture cells we are handling, or whether we are handling with viruses. That is also important. So basically, this airflow is continuous and maintains somehow sterile or sterile-like conditions. If you have any interest, there is a link in the bottom of the slide for additional information on different types of hoods. So an important thing is that, so you are used to the pipettes in the bench, but in the cell culture hood, we also use different style of pipettes. This is because we are handling large volumes of media. And so we have different types of serological pipettes, the same way you have different types of tips for handling different volumes, with the great help of the automatic pipette. Then we also usually have a vacuum aspirator, which allows us to aspirate medium, and that goes into a liquid weight container. Other essential equipment that you might be familiar with would be a centrifuge. That would be very helpful for cell collection. Of course, an inverted microscope, which will allow cell monitoring at any stage. It's very important when you are culturing the cells to keep a close eye on them. You don't need to check them every hour, but certainly every few days, depending on the cell type. As you also may know, cells live in a humid CO2 incubator, and when we want to freeze them, then we can cryopreserve them in a liquid nitrogen. Another essential piece of equipment, it's a temperature-controlled water bath. As some of you may know, you need to warm up all the cell culture medium or all at 37 degrees. Okay, so before we delve into some more parameters, of course, the first important thing is to think that we are culturing live cells. So it is really important to follow some sterile technique rules. And so the same way we need to wash our hands nowadays and disinfect things, this is our regular modus operandi in the cell culture room. So wearing gloves, it's recommended. And in any case, very important is to disinfect with ethanol the hood before and after using, and anything that goes inside the hood. And then all the medium and reagents that we are using, if they cannot be autoclaved, because otherwise they'll be damaged, then we need to filter sterilize. So the idea is that we keep a set of reagents that are only used in the culture to avoid cross-contamination. And then another thing that you will see later in the video is that it's also very important, the sterile handling. So not using the same pipette over and over in different mediums or in different reagents. So that is all together what allow us to not contaminate our culture and the cells to grow healthy. Okay, so going back now to real cell culture, thinking about you are about to culture cells. So of course, the first or the most important thing is to think, that you need to recapitulate in the dish in culture some of the important in vivo growth requirements for the cells. And of course, each cell type will have a different function in their residing or tissue origin, and they're going to be surrounded with a very specific microenvironment, which will have unique physicochemical properties. So of course, when you're thinking of culturing a cell, you need to keep this in mind to be able to define which are the essential ones that you need to then recreate in in the dish. So for example, the inner cell mass, where we isolate ES cells that you'll already heard in FibroMass lecture and you'll be hearing throughout the day. It's very different environment than the skin, for instance, where fibroblasts can be isolated. And these two are really different environments from the neurons that reside in the brain, for instance. So all of these together, you need to keep in mind. So each cell type has different functions and different growth requirements. And that means, of course, that they have different morphology, for instance. So you can see here an ES cell colony. The cells are very compact and growing together. And if you compare to the surrounding fibroblast that have a spindle and elongated morphology, look very different, for instance, from these neurons. One more thing that is really important is that once you are culturing your cells, it's not just to keep them alive or keep the morphology that they should have, but also they should maintain, or the goal is that they are functional. So for instance, you can see in this myocyte culture, they still were able to keep their beating capacity. Or for instance, these firing neurons. Look, using a calcium dye, you can see their activity, and we can record all of these activities. So each cell type has different growth requirements, some growth in a monolayer in adherent conditions, but others grow in suspension. So we'll go through some essential parameters, which are the cell density, the substrate in which they grow, the surface area, and the culture media. So of course, cell density matters, because if you think a cell receives signals from the neighbor cells, either in secreted form or in contact-mediated form. Of course, it's not the same if you have a single cell in the dish that you have a dish full of cells. So once they proliferate, they do so until they reach confluency. That means when the dish is full, and then they stop growing. So one really essential parameter is the substrate you choose, and that you can change its chemical composition. So some cells grow already on directly on plastic, or you can use different types of polymers or different types of extracellular matrix molecules, or a combination of some of these. You can also change the physical composition of the substrate. It can be liquid or solid, but you can play with the stiffness of the substrate. And it has been shown that the same cells behave in very different ways, whether they are in soft or in stiff conditions. So for instance, thinking about later on your experiments that you're going to be doing virtually, so the same cell on a substrate, on this blue substrate, for example, will start proliferating. But the same cell in a totally different substrate will no longer proliferate. Instead, it will start differentiating. So really, the choice of substrate is going to determine the output of what your cells will be doing. So as I mentioned earlier, so the cultures can be in very different sizes. So you can culture in a well, just one cell, or you can do up to millions of cells. So, and this is possible because we have many different vessels where we culture them, and they all come in different sizes: dishes, flasks, well plates, chambers, and all adapt to what your culture needs. So the environmental factors are also very important. As I told you, cells are cultured in a humidified incubator, and this provides constant temperature, which is 37, is the body temperature, but also constant CO2, O2 levels. So usually cells are grown in 5% CO2 and 15% O2, oxygen. Of course, many researchers use these parameters as experimental parameters, and it has been shown that, again, if you change the CO2 or O2 levels, cells also grow in different ways, and the behavior it's altered in very different ways. And finally, the most important of all, it's the cell culture medium. So I would say that most cells are cultured in basal medium, that's DMEM, which provides basic nutrients, osmolarity, and it very importantly provides buffering system. So you might probably you have seen that most mediums are red, and that's because they have phenol red, and that's a pH indicator. And that's very important, you'll see, because it can indicate you the status of your culture. So basal medial is the basal composition of all mediums, and then each cell type will have a specific and unique set of growth factors or supplements which will be required for their growth, their maintaining their function in the dish. For instance, you'll be seeing ESLs have a very different growth requirements and media composition than, for instance, neurons. And then in addition to the sterile technique that we mentioned earlier, some cells are also grown with antibiotics or antibiotics, basically so to avoid the growth of bacteria or other types of contaminating agents. So finally, so each cell type has a different growth rate. And this is really important because when you are culturing a particular cell type, then you need to know what's their usual division growth dynamics. So an important aspect that you'll see in the video is that passaging the culture needs to be done before clone fluency. If you leave it for too late, let's leave it for tomorrow, maybe your cells will start differentiating or dying or not dividing anymore. So finally, a very important aspect is that when you do the passaging, when you split the cells, this has to be done at the right density. And again, this is unique for each cell type. So then once you're getting used to your culture, each cell type will have different requirements. So we thought after this brief intro to some of the important parameters in cell culture, we thought maybe it'd be nice, especially in this virtual form of the experimental course, to give you this video of how to passage cells. So also to get a flavor on what it is to work in the hood and with cells. So then let's watch this video. I will share the screen. If someone has any problems, please let me know. And after the video, we'll have a round of quiz questions with the Kahoot. And also if you have other questions, we can answer them at the end. So write them down and we can discuss them in a bit. Okay. Okay. So I've been told that nobody can everybody hear or someone? No?
I hear nothing.
Okay. So I will start again. I mean, just bear in mind with me 1 second. Okay, we'll try again. Okay. Can you all see the movie, or at least the image? Yes, okay. Can you hear? Can you hear? Can you hear anything?
Yes, now we can.
Passaging or subculturing of cells is a common procedure wherein cells rendered in culture are divided or split into new cultures and fed with fresh media to facilitate further expansion. While the concept itself is relatively simple,
We can't hear anything again.
It might be that when you mute yourself, the audio disappears. Because it was working for like half a minute and then stopped.
So we're still not being able to hear the sound. So maybe you have the link at the bottom of the page. In the last slide, can you see it? Yeah, so. I am not mute.
definitely. The famous immortalized cell line is the HeLa cell line, which was derived from a cancer lesion found on a.
Can you hear that? Okay, so okay, so they need to work. Okay. 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 So let's try, otherwise I'll send you the link. Yeah, sorry about this. Share computer sound. Okay, so thank you. So no, I don't think it's possible, actually. So let's try this, otherwise. Can you hear? No?
Passaging or subculturing of
Now,
cells
now we see the wrong screen though. We see your desktop.
driven culture are divided or split into new cultures and fed with fresh media to facilitate.
So you don't see the video?
No, but we hear the sound.
Okay, so that's
I mean, if the sound is just completely not working, you can just play the video because the video has subtitles. So we can read the subtitles even without the voiceover, I think. Or if you can't sync it up properly.
Further expansion.
Can you see the video?
While the concept itself is relatively simple, in this video,
Can you see the video now?
Like only the upper half of the screen.
Yeah, but you can see that screen. Okay.
we'll discuss some of the more important steps for the successful maintenance and expansion of cell lines.
Okay.
Toxic metabolites accumulate in cell culture media over time. When expanding cells, it is particularly important to change the media regularly to maintain cell health and to monitor the cell expansion to avoid cell overgrowth. Generally, two main types of cells are subcultured: immortalized cell lines and stem cell lines. Immortalized cell lines are cells that are derived from multicellular organisms that have experienced some type of mutation affecting their cell cycle regulation, allowing them to proliferate indefinitely. Perhaps the most famous immortalized cell line is the HeLa cell line, which was derived from a cervical cancer lesion found in a biopsy taken from Ms. Henrietta Lacks in 1951. In contrast to cell lines in
Can you still hear? Can you still hear? Can you tell me?
No. It was working before.
Okay. So, so before we go for, if you have any questions, we're gonna have a little game with Kahoot. So I think you've already had an introduction session by Luca. So so you need to go to kahoot.it and I will give you the PIN number to answer some questions. Just hold on 1 second. So everybody, it's okay with Kahoot? Yes? Great. So I'm gonna need to share my screen. Okay, so this is the PIN number. Can you all see? Okay, players coming up. Few more people missing. We are missing some people, so if you could put the PIN number, so unlock. We're still missing some people. Hello? So Okay. So there are 39 people. I will start. I think there's still few people missing, but yeah, maybe we can catch up later for some of the concepts. Okay, so ready to play? Okay, first one. When do you need to split your culture? So when it's 80 to 90% confluency. Remember not to wait until it's really full. Okay, Talia? So one reason why plastic cell culture containers are commonly used. Okay, most people, yeah, many adherent cell lines naturally attach to uncoated plastic. So what does it mean if your culture is yellow? So yes, the media is acidic. So remember the phenol red in your culture medium shows you the pH and so the status of your cells. So substrate is only important for so none of the above, because it's not just for one of these, it affects every process or behavior of the cell. So now Sandra, it's up. Well done. The cells are growing best at so remember, each cell type is a different world, so has unique requirements and grows at different rates. So you'll have to know for each cell type. Okay. Pius is on the top. So which are the essential parameters in cell culture? So yeah, cell density, substrate, and culture medium. All of them are essential and very important. Okay. Talia, Pius, and Simona. So well done, everybody. Okay, so I don't know if you have any questions, would like to ask, maybe you can speak up or type them. You're ready for the next one? Yes, okay. So now we're gonna have a short break and then we meet again. So I think the idea is 11:15 and then you need to contact. So I think each tutor will have sent you a link for the individual tutorial Zooms. So we're now gonna break into smaller groups. If someone has any questions, can email us in the meantime. Okay, bye-bye.